Okay, we are about to start one of the most complex things in living organisms and the study of biology. We're doing a unit on photosynthesis. It's actually going to be in 3 separate video lectures. First one today is going to be a basics on the biophysics of light. It's kind of the precursor to understand all the complex reactions we're going to see. And then we're going to spend one lecture on the light reactions, which is the first step or process, and the third lecture will be on the Calvin cycle, and which will be the last step. So we're looking at biophysics of light. This is going to be just the most basic biophysics thing you'll ever hear, but to me, if you don't understand this, then you're just kind of memorizing things in photosynthesis. And as people who want to go into the sciences, we need to understand this. So first of all, we need to know where is energy originating from. And it is coming and streaming through the universe from the suns and the stars in all directions. And as it's moving through space, it hits an object. It's going to, these thermonuclear reactions, about a third of it is immediately reflected back into space. Most of the rest of that energy that hits Earth is in an unusable form. No organism can use sunlight to fuel their metabolism. And so most of the rest of it just heats the Earth. Less than 1% of all that energy, and my number here is it's approximately equal to the amount of energy that hits Earth each day, about 1 million Hiroshima-sized atomic bombs. That is how much energy. But a third of that's reflected back out to space. Most of the rest is absorbed by the Earth and heats the Earth. And less than 1% of that energy that comes in this form of light hits a photosynthetic organism that can convert these energy in the form of a photon into sugars. And now once we have it in a chemical, from light into a chemical, now it can be used through the organism that did it. Other organisms can eat it and they can get that chemical, and it can be stored as well. So let's look at the unique things. We know that this energy is coming in the form of light from the sun. And light has 2 types of properties. It has a wavelength. Each different type of light has a different wavelength. You can actually measure crest to crest like you would a real wave, a sequence of waves. The distance between each wave is the wavelength. Each different type of light is unique. And then they also have these particle-like components or characteristics, and those are possess discrete particle-like units called photons. They have a discrete packet of energy. And what we find is that the particular wavelength is associated with a particular type of energetic or energy in the photons of those units that make it up. And what we see is that it has an inverse relationship. First, let's look at the types of light, with gamma rays having the shortest wavelength, and then X-rays just somewhere between 1 and 10 nanometers. We measure intracellular structures in nanometers, so it gives you an idea of how short those wavelengths are. And then there's this little tiny bit here, don't forget ultraviolet light, and then there's a little area here which we call visible light, because that's the range of photons that human eyes can capture. A little longer than visible light or infrared light, and then radio waves are the longest. Some of them are hundreds of meters long. And if we look at the visible light, we see the blues are on this side. They have the shorter wavelengths than the reds that have the longer wavelengths. And then if we look at how much energy, again, the relationship is the longer the wavelength, the less energy the photons have. The shorter the wavelength, the more photons. So if we look at gamma rays, we see they're the shortest, then we know they have the most energy in the photons. And radio waves, longest, they have the least energy. Just so you can kind of really get that, we're just going to look right here at visible light. And between this wavelength, 400 and 740, there's energy in here. What color do you want your Coleman stove flame to be? You want it blue, because there's more energy in the photons and it will cook. Then let's think about this. Let's say you're going to go into the classroom and it's March and you're going to get your bikini on and lay out on the tables. Is there enough energy in photons of visible light to do any kind of damage to your skin from that light bulb? And there is not. Right? We could lay there for days. However, now let's go just a little shorter. And if we look at sunlight, it has a much higher proportion of ultraviolet light in sunlight. And now on a March day, let's take someone who's as snowy white as me and take them down to the equator. How long would it take for that UV light with just a little more energy to damage my skin? It wouldn't take me 10 minutes, okay? So just this little bit difference in length, a huge amount of difference in the energy in the photons that make up those wavelengths of light. All right, so as this energy, again, is bombarding the Earth or anything that it hits, the light has one of 3 things that can happen to it once it hits any kind of object. So let's say this is the ray coming in. It can be reflected. It can be absorbed. Now, when we say absorbed, it's actually going to be used molecularly. And when it uses that energy to do something, it's gone. It's just like if you have a cake and you eat it to do something, make energy, it's gone. You don't have a cake anymore. So when light is absorbed or captured, it's gone. It's used for something, so that energy is then gone. Or it could be transmitted. So it could be transmitted some other place, okay, for something else to use. So anytime light strikes an object, it can be reflected, it can be absorbed or captured, or it can be transmitted. And the molecules that can absorb or capture light, we call those pigments. And molecularly, they are arranged so that they can use the energy in the photons, and they boost electrons to a higher energy state. So once they use that energy to move them, that energy's gone. Like if I use that energy of the cake to run 10 miles, the cake is gone, right? So these molecules that have this ability to capture photons of light and use them for something are called pigments. Okay, chlorophyll, flavonoids, carotenoids, anthocyanins. These are all some examples of molecules that can actually capture the energy from photons of light, which is just what I said there. As we move into photosynthesis, we're going to look at two major types or classes of pigments in photosynthesis. The major primary photosynthetic pigments are those called chlorophylls. Chlorophylls actually consist of chlorophyll A and chlorophyll B. And then the carotenoids, also known as accessory pigments, as well. I think I've got a sticker on that. Here's a molecular structure of chlorophyll. Notice it has these ring structures, the porphyrin ring. Usually has to do with fire, porphyrin. Hydrocarbon tail. It's these ring areas that when during photosynthetic process, the electrons are going to capture so many photons of light, it's going to be excited and kick off electrons, oxidize. Again, two major classes of accessory pigments. Well, the major one is carotenoids, but there's also these phycobiliproteins that are important in low light areas. Some examples of carotenoids, again, the flavonoids, which actually is kind of a sunscreen for plants, xanthophylls, beta-carotene, the anthocyanin that we looked at earlier as well. I like looking at this. This tells me a little bit about the wavelengths of visible light. Here's the blue, here's the red wavelength. And it shows the carotenoids capture some of the blues. That's it, okay. And then wavelength, the photons in the wavelengths of light. Chlorophyll A gets most of the bluish violets, and then it gets a lot of the lower end of the red, the photons in the red wavelengths of light. Chlorophyll B, shown in the dotted line, captures a lot of the blue and less of the kind of the orangey reds. So what this means, now you're thinking, yeah. But if between all these different pigments, these wavelengths of light are absorbed, captured, the photons are used by the plant for photosynthesis, right? So cover that up on your screen. And then over here with the oranges and the reds, all used, captured by the plant pigments, used, gone. And if you cover those two areas, what's reflected is the green and the yellow. And that's what bounces back and hits our eye. And this is why we see plants as being green, because our pigments in our eye can capture the green wavelengths, and the plants reflecting. They're not using those wavelengths of light. That's why they're reflected, but our eyes can capture them, and we see them as being green. So I'm wearing a blue shirt. The pigments they added to the cotton captures the greens, the yellows, and the red wavelengths, all gone. Blues are reflected. The pigments in the shirt does not capture those wavelengths of light, those photons, and use them. And so they reflect, and they hit your eye, and you recognize that wavelength of light as blue. Cool beans, huh? All right, so then in the fall, right, again, in the regular time of year, the chlorophylls are the primary ones, and they're capturing the blues and the reds, and they reflect that green, right? Green wavelengths of light, not colors, wavelengths of light. But I do want you to see this. In the fall, and there's a couple different hypotheses, and I'm thinking it's probably going to have a lot to do with the species, perhaps, of plant, but the light comes in a little different angle, and then it gets cooler at night. And it does seem that the chlorophylls break down when the nights have been cooler, like in the 40s, over, I think it's a 7 to 14 day period, pretty much straight through, they break down sooner. And so these accessory pigments stay around longer. Notice they're capturing the blues. So once the chlorophylls are no longer capturing the reds, and these guys aren't either, then this is what you see reflected, the reds. And this is why we see these plants as being red or yellow, depending on which accessory pigment they have and which wavelengths of light they're capturing. All right, the last thing we're going to look at is some leaf and chloroplast structure. I want you guys to have this completely mastered. Everything from biophysics of light, from ATP energy, and this leaf and chloroplast structure has to be completely at the top of your head, ready to speak without hesitation. Otherwise, the lecture on the light reactions is going to feel like you've dropped in from outer space, and it's going to feel very foreign. So make sure you've mastered that. If you don't have these mastered, when I start talking about this happened there, it's just going to be a big jumble. All right, so make sure that's mastered before the next lecture. And if we're looking at this leaf here, this would be the upper epidermis, these cells. This would be the lower epidermis. This would all be the mesohyl in here. If we take one cell out, we see it's got the walls, nucleus, central vacuole that we talked about, and a lot of these chloroplasts. In fact, a real one probably has a lot. I mean, when we watch them we look at Elodia under the microscope, and they're just moving around, just tons of them. All right, here's an electron microscope photograph of this, but here's what I want to point out, because as we break down these different steps or processes of photosynthesis, they are occurring in different places. So each chloroplast has two membranes surrounding them, this inner membrane and this outer membrane. And then there's a set of membranous discs or vesicles. And so they're like membranous sacs, and they're stacked. These are the thylakoids, also called, sometimes some folks call them thylakoid membranes. And then these are hollow in the inside, so there's a lumen or an opening. So again, imagine a membrane, and you've got this empty space in the inside. And then notice these are generally stacked. These thylakoid are stacked. Those are called one granum. For referring to plural, it's grana. And then all the fluid outside of that, so all the fluid in between, all those thylakoids, all those grana, all the fluid that fills up the rest of the chloroplast is called the stroma. The stroma. We're going to see that the first step of photosynthesis occurs on the thylakoids, right here. And then the second step, Calvin cycle, occurs in the stroma. So again, we want to make sure that all these steps are mastered. No hesitation in your learning at all before you start the lecture on the light reactions of photosynthesis. Good luck.